Hello all, this is Dr. B. This is a tutorial on how to use Uniprot. Uniprot is a collection of proteins, annotations for the proteins, etc. And it has four basic, for lack of a better word, compartments to it. So the first one is, we'll just start here with the Uniprot knowledge base. This is also called basically Uniprot KB. This is the data of the functional information on proteins. And there's two different ways that information about the proteins has been basically annotated. One of them is through something called Swissprot, and the other one's called Tremble. And Swissprot is the one that's just basically a manual annotation and review. So this has been reviewed by, for the most part, humans. I don't assume AI does it yet. And then there's unreviewed, which is Tremble, and this is automatically annotated and not reviewed. Remember, annotated basically means kind of like identifying a particular either domain or function or sequence of a particular protein. It's basically form fits function. So that is the first one. The second one, basically called the proteomes or species. This is a set of the proteins with complete sequences of organisms that have genomes or complete genomes. So basically, it's thought to be all the proteins in organisms with genome sets. Then we also have something here called protein clusters, and this one's more commonly known as UniRef right here. And basically, this is more about identity, and so it's proteins that have a fair amount of identity. So if you have, a mystery protein, that's what you would use in essence to be able to find out what might be other homologous or other proteins. I'll just take that word out for a second. That might be related based strictly on identity. And then there is an archive of all of these across different databases. So this is just a generic, just kind of view of it. A lot of this intro also is just for later on, as well as the so in terms of the application. Sorry about that. So there's a bunch of different things you can do. You can search through any of these: Uniprot KB, Uniref, and we'll talk about again when we would use some of these things. We can do an advanced search as well, where we can look at a particular gene name in a particular organism with a particular keyword, which is really nice. They've updated this from the older version. And again, some of the things up here you probably already know, like particularly like BLAST. So like in terms of when we would use these here. So BLAST would be for finding, regions of similarity between sequences. And the alignment here, if you were to do this, would obviously be aligning, two or more sequences. It uses cluster omega, I believe. And then we have the peptide search, which obviously is a list of all the peptides to find Uniprot sequences that have them. And then we have this thing called ID mapping, or it's also used to be called, I believe, retrieve. It's a list of identifiers to retrieve the Uniprot entries and map them to some sort of database that's outside of Uniprot. And then we also have this cute little word here called Sparkle, and we're not going to be using Sparkle, but basically it's kind of complicated, but I like to look at it like you can combine data from a bunch of different sources to create kind of like your own database, and you can use Uniprot data with other pieces of data, a lot of times written in languages similar to R, if you're familiar with that. But that's kind of what it is. It allows you to go a little bit farther and do cross references of databases along a lot of other options. So we're not going to really spend a lot of time on that. So let's get into it. All right, so notice, by the way, it defaults right now to Uniprot KB, the knowledge base. So let's just do a couple things here. Let's just say we want to look up a protein called ADE2, ADE2. And that's a protein that I'm interested in. Let's just say I'm interested in. I want to know about its activity and its annotation in yeast. But let's just click the search and see what we see. And we're like, oh, geez, there's a lot here. So the terrible way to try to do that is to look down the list of the organisms and try to find Saccharomyces cerevisiae, by the way, which is baker's yeast. Oh, there it is. So I could have done it that way, but there's a much easier way to do that. If I already know more information, then I will just do this. Add 2, and then I can go down to either of these, it doesn't matter, and I can go to organism. And as you'll notice, I start typing, it's like, oh, you mean Saccharomyces cerevisiae, baker's yeast? Yeah, that's what I mean. And there we go. And so that's it. That's the one I want to know about. And you can see that this is 571 amino acids long. And let's just click on the entry itself. And you can see here that on the left is a series of menu items, some of which are highlighted, some of which are not. And basically, that's partially if it's not highlighted because there isn't information in that particular category. But they do go in order. So I can see here its full name is, look at this, phosphoribosylaminometazole carboxylase. I know what it does, but wow, that's a funny word. And so there's if we scroll down, we can see that there is some information about it. There's the basic kind of what it does. And as we keep scrolling down more, we can see that there's all kinds of other, like what its function is. We can see some different functions or processes that it does. It looks like it's a decarboxylase. And as we keep going down, we can see other names, the gene name, the organism. This one, it's la 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 la, down the line, family and domains. So that we're going to talk about domains in a little bit. And it shows you kind of the particular domain databases of where we can view some of these things. Here's the actual amino acid sequence, and even more information. Again, you can see, notice the difference now. You can see we have our Swissprot versus our Tremble, and obviously this is a little star there, so that's been reviewed. So I'm just going to take a little quick pop onto that and see what that is. Like, oh, okay. So this happens, notice it's the same gene. This is stuff that has been reviewed. And you'll see here that this particular publication, which has been peer reviewed, is probably the source of a lot of the data that's down here. And what I really like, by the way, is this. It can kind of show you how this works and kind of what it does, which is really cool. So if I click on the left, I have names and taxonomy. Then I have subcellular location. And you can see this is a cute little budding yeast. And if I just kind of scroll over it, it shows me that this is produced in the cytoplasm. So that's really cool, that it can show you where in the particular cell that any particular protein is actually present and or produced if we know that, and which is really cool. If we get down to phenotypes and variants, you'll see here that this one didn't show it from that view, but once I clicked, it did show it in this. And there's not much here because this particular protein doesn't have that annotation to it from this particular database submission. Then we have PTM, which is post-translational modification. And there's not much here because it's very simple. And I'm going to now show you a protein in a minute or two that's not simple. The one that does have a lot of different modifications to it. Also, what this thing interacts with, and you can see there's a structure, which is really cool. There's a bunch of ways that I'm turning it, by the way, which is really neat. And people that understand proteins, this will make sense to them. You have some beta sheets up here in the back, and you have some alpha helices and a bunch of other cool stuff. So if we keep going down here, once again, we've already done interaction. There's the structure, family and domains. Again, not much information on this sequence. So the reason I showed you this was just kind of give you a sense of what you can see from like the left side about any protein, which is really nice. So let's try one that's a little more complex. Let's do P53, tumor suppressor protein. But once again, you'll notice that there's a whole lot of samples here. There's a whole lot. And so you're like, oh, where's human? Again, I could go down here and choose human, but the easier way, I could have done the advanced search and found it that way. So let's just assume now I get to this. So let's take a look now at this. You'll notice that there's a lot more information about its function. And these are all of the different, this is almost like, in essence, a citation. All of these things are the particular, what is this? A double-stranded repair protein involved in DNA damage. Well, how do we know that? Well, it came from this particular paper right here. And so that's kind of what that's talking about. So, excuse me. So now we have some annotations, and this is gene ontology. And from here, notice how it's like, whoa, what's all this? I'm not going to get into this part now. I'm going to get into it later. But these are some gene annotations that have been done on this particular protein, and I'll get to that part later. So if we just do a quick kind of tool down again, we see the gene name, we see the lineage of humans, which is where it's found. We go down to subcellular location, and what do you notice? That this is the area where this particular thing is produced. That makes sense because it does interact with DNA to some degree, and because of DNA damage. And how do we know that? Well, you'll notice here, these are the publications that showed us that that was the case. And that's why this is such a useful database, is because it has so much that is very easy to identify how we know the information. Very much unlike the internet, where you have no idea who wrote what and what's fake news and what's not. It's very difficult for this information to get in here. A lot of steps have to happen, so it ensures that what you see is accurate. So that's a really neat thing. And now we talk about this. Now this is going to be a little different than the add to, and you'll see here that like what is all this stuff? Well, this is basically mutagenesis and or areas where there tends to be some sort of mutation or difference. And some of them, again, is a mutagenesis, and some of it is a natural source. So if I just kind of hit one of these, we can see that this is a particular area where there's been some mutagenesis, as is this. And so you can see these are all the different areas where there's shown to be some sort of difference in sequence. Again, here's some more mutagen. So that's just, I don't want to overkill, beat the dead horse. So we move down. Let's do some post-translational modifications. So you'll see here that this also has other areas of modification that happened after the DNA is transcribed into RNA. And there's a whole lot of them, and you can kind of see from there that's a whole mess of them. And as we keep going down, we see what else this thing binds to. What other interactions does this thing particularly have? And that's another great thing as well, is if you want to find out what does P53 interact with, this will give you the answers to that, and here are the publications that kind of align it and show it. As far as the structure, this will probably take a second to load because it's a pretty big protein. Well, that wasn't too bad. So this is really neat. You can see where I can zoom in and zoom out. I'm just scrolling my mouse, and I can flip it around, and I'll do all kinds of stuff. What else is good when the proteins are a little more complex? This is a quaternary protein, actually. It has 4 subunits to it. Is that I can actually identify exactly where something is, and you can see I'm scrolling over this, and it's showing me the bottom right where I am on the actual protein. There's serine number 111, and as I go along, I can alanine. I can go through all of them, which is really cool. This is very useful, especially if you're interested in some sort of where a mutation might occur. So these are some of the features of what these things are. So amino acid 1490 to 1494 is a beta strand, a turn, a helix, and so this is literally related directly to this. That's all related back and forth. If you wanted to see how a particular protein might be affected by its mutation. Now you'll notice that this has a lot more domains to it, and a lot more regions, and they do all kinds of different things. And you basically could expand upon these, and see which ones they do. And there's a whole bunch of different information here. Again, this is just an overarching view of this. We're going to go into a little bit of detail later. Then, of course, now we have the amino acid sequence, and there are 3 isoforms. And isoforms are basically versions of proteins that differ because of improper or different RNA splicing. And so that basically is showing the 3 of them. There's the 3 right there, and you could even compare them if you wanted to do it by aligning the 3 of them, and maybe looking for where the differences are between some of these different isoforms, like what might be missing, what might not be, etc., like that. I'm going to pause the video for a second to let this load. So that took just another minute or so. So we can see that there's a little bit of difference between these 3 isoforms. Let's see if there is any more. Oh, there is. So you can see here that there's definitely another version. So again, I'm just giving you a generic kind of like WebQuesty view of a lot of these, so that you can just kind of get a generic sense of kind of how these 3 operate together and what they do. You can also create phylogenetic trees with it. I'm going to kind of get back to where I was. I don't want to go too far down the rabbit hole at this point. So continue with the P53, we see that along the amino acid sequence, there's all different kinds of families, features, domains, etc., all indicated here. And again, we looked at the sequence and isoforms. And then there's, of course, the idea of similar proteins. So you'll notice that there are no similar proteins at 100% identity. But then what I can do is I can see, well, are there any at 90%? And the answer is, well, yes, there are. And the reason it's showing up the same protein, if you recall, is because of the isoforms. And that would, obviously that makes sense. But we also see that it's very, very closely related to other organisms who have relatively very similar, and this makes sense. You would expect to see the chimpanzee have a very, very similar one, because of the obvious relationship between humans and chimpanzees. So this is just like a kind of starting point of how to look at Uniprot. I'm going to go basically through the top as well. So there's a couple other things. The variant viewer I showed you before. This gives you more of a 3D kind of view. But now notice how different this looks compared to the other add to. And what I can do here, you can see, is I can choose to look at whatever I want to basically look at. So I can choose any of these. So predicted sequences. We can look at anything that's likely pathogenic, which of course there isn't any. Excuse me. And so we can see where the variation occurs. So we can see that these are the different amino acid changes that we see, all the different variations that we see. So that's a little bit of that. Feature viewer is going to give you kind of an overarching view of everything. I really like this one, of everything from process. By the way, everything is here that you actually see. Sequence information, domains, we can turn them on and off. We can collapse them. I like this because of its pictorial view, because you can overlap more than one thing against the same amino acid sequence. So you can compare this area right here and see what different things it might have. Which is really cool. And of course, it gives you whatever region this would be, and you can look at all of the different annotations that are there, which is really nice. And you can see how it's just kind of like a folding, make it shorter, longer type thing. Publications, here's basically all the publications that have been indicated in the paper, if you want to look at it that way. So that is just the first video of an, oh, I know this is my longest one of the course, but just to give you a view of this. So I'm going to have you just do one little activity where I'm going to give you an amino acid or a protein, and you're just going to explore and tell me some cool information about it. Then we'll move into a couple deeper things that Uniprot can do.